I'm from the University of Minnesota in the School of Public Health, and also representing our project supported as part of the Upper Midwest Agricultural Safety and Health Center, which is a collaboration between the School of Public Health at the U, the College of Veterinary Medicine at the U, the State Health Department, as well as Marshfield Clinic in Wisconsin. And I want to recognize my co-authors. Etta Peju. Edicina is a doctoral student with me in the School of Public Health. And she has a poster, or had a poster last night, up related to part of this research. Dr. Abu Bakr is a postdoc in the College of Veterinary Medicine. Mi Yang is a research scientist who works with Dr. Tor Morel. And Dr. Goyal is also involved in this project. So kind of a motivation, this, the Upper Midwest Agricultural Safety and Health Center, UMASH, is funded by NIOSH, which is the National Institute for Occupational Safety and Health. So they are interested in diseases that affect people. So we're interested here in diseases among animals that can affect not only the animals but also people. So we're focusing on influenza viruses here. Although the work that we're doing will also presumably be able to be used with viruses like PRRS and other viruses that affect swine. So we know there's emerging zoonotic influenza viruses that pose health effects, either real or potential, to swine workers, poultry workers, veterinary workers. And many of these viruses we know can be transmitted through air. We don't know how much of that transmission may occur through air, either among animals or between animals and people. Or those viruses have the potential to develop airborne transmissibility. Animals in agricultural facilities generate virus-containing particles small enough to be transported substantial distances. So that's an important factor to keep in mind. And also, little is known about the typical sizes and concentrations of these particles that contain viruses in animal agriculture. Or, importantly, if those viruses remain infectious once they are airborne. So just because the viruses become airborne, that doesn't mean they're necessarily going to be infectious. As they travel through the air, become dehydrated, they're likely to lose their infectivity. But we don't know the extent to that, and that extent may vary depending on the type of virus or even the strain of virus. So the other thing that we're interested in is particle size, and that was alluded to by Kevin earlier. And so why do we care about that? Well, one reason that Kevin talked about is our ability to collect different size particles is highly variable. So that's an important reason to know the sizes of particles. That's my third reason. First of all, we want to know how far those virus-containing particles can potentially travel. Larger particles are going to fall out of the atmosphere more rapidly than smaller ones will. So that size matters as far as ability to travel through the air. We also want to know potentially where the particles may deposit, either in the respiratory tract of people or of animals. So if they are smaller particles, they can be inhaled deeper into the lung, maybe cause different likelihood of disease based on that. Or they may not be able to be cleared as well if they're smaller particles. And also, as I mentioned, the ability of the particles to be filtered or be controlled by some other type of technology. So our research objective is to measure better than we do now what is in the air as far as virus-containing particles. So we want to either identify or develop a high-volume, field-portable, size-differentiating viral aerosol sampler and use it to measure worker exposures to live airborne influenza viruses in animal agriculture facilities. So key points here: viral. We're working with viruses, not other types of microorganisms, at least not at this point. We're interested in particles that are airborne. We want to collect samples from the air so they can be analyzed. We want large samples, large volumes of samples, with the idea that that'll allow us to have a low limit of detection on our analytical procedures. So we really have a good chance to catch what might be there in the air. We want to do this in the real world, not just in a laboratory, so we want it to be field portable. We already talked about the size differentiating importance, but that's something we want to get out of this. We want to know how much of the viruses are actually infectious, if at all possible. And then our focus is on animal agriculture, as opposed to other types of occupational settings where viruses are important, like human health care, although this type of device may be useful in those settings as well, if we're able to identify or develop one. So there's a lot of factors that we're trying to think about at the same time as we develop a sampler. So our first step in this, and we're still only on the first step, is to evaluate existing samplers. So we want to first assemble a wide range of existing samplers that operate by a variety of principles. And test those samplers side by side in an isolation room using mechanically generated aerosols. So we're doing this in a semi-controlled setting to do this comparison initially. And use those results to determine combinations of sampling parameters and technologies that give us the best chance of collecting viral RNA as well as live virus. So let's talk a little bit about the sampling technologies that are available. So first, and these first three drawings are from an article by Klaus Willicke from 1998. They're not my own drawings. So the first technology is impingement, which basically is an airstream comes in carrying particles, and this is usually an aqueous solution of liquid here. And the particles come in, and the air flows, and it becomes all frothy. And the particles are collected in that frothy liquid as they move around by sort of their inertia bumping into the liquid as they bubble through and get captured by the liquid. So that's impingement. You have cyclones. So particles have inertia, and as an airflow comes in and it's given a sort of a circular path, it's actually a helical path, the particles that are large enough diverge from the airstream lines and impact on the outer walls of the cyclone, or the outer inside wall, if that makes sense. I'm not saying that quite right. And can be collected on the inside wall here. So that cyclonic motion, sort of centrifugal force, allows you to capture the particles. You have impactors. And as air comes into an impactor, the airflow turns 90 degrees. And again, some particles, if they're big enough by their inertia, can't make that turn, and they impact on the surface and can be collected. You also have sampling filters. And this is just a little drawing that shows a filter fiber. And the reason filters work so well is that they can capture big particles, just almost like sieving some of them, but also by their inertia, as they deviate from airstream lines, they're too big to go around the fiber, and they impact over here. And then the littlest particles move around by diffusion, and they bump into the fibers. So filtration can be really effective for both big and small particles. You actually have the hardest time capturing kind of the in-between size particles, although filters can be designed to have any efficiency. So sampling filters are also a useful technology, not only for controlling particles, but also for sampling them. And then we even have electrostatic collection, where you can ionize the air through a wire, and these ions flow out away from the wire, bump into particles, and then if you have a surface, not shown in the picture over here, then the particles, that surface can be grounded, and the particles can move towards that surface and be collected there. And there are also combinations of these different sampling technologies that can be combined into a single sampler. In particular, impingers and cyclones often go together. So you can have something that has cyclonic motion, while at the same time that cyclonic motion can be going through a liquid. So you can have both the cyclone technology and impingement technology at the same time. So we assembled, I think it's 16 different samplers for our tests. And there's others that are out there that we've identified since then. And there's other types of the same technologies that may be made by different companies. But these are the samplers that we settled on for our tests. We have three groups. The first group is primarily based on impingers or cyclonic impingers. The second group is mostly filters with one electrostatic sampler. And the third group is focused on impactors. We did keep two common collectors in all three sets, ones that we've used previously, both in the laboratory and the field, an Anderson impactor and a cyclonic collector made by Midwest Microtech. And then in group one, we have a couple of different types of impingers, an AGI 30, which is an old or has been around a long time type of sampler, a biosampler, which maybe has been around for 15 years. NIOSH, the federal agency that I mentioned that actually is funding us, their staff developed a bioaerosol sampler that can be worn by a worker to measure their own exposure to viruses in 3 different size intervals. So we tested that. We, based on from a company called InnovaPrep, we borrowed a couple of their instruments that are high volume, a SpinCon 2 that is a cyclonic impinger, and then a Bobcat, which is primarily a filter-based sampler. So it was included in this group because just of the timing of the tests. And then the Vivas is a sampler into an impinger that is developed by the University of Florida in combination with a company named Aerosol Dynamics. The second group, we have several different types of filter samplers here. Particularly one that's different than the others is the high vol sampler, which these were sampling these 3, one of which had a sampler inlet to cut off some of the bigger particles. Sampled at relatively low flow rates. This high vol sampler sampled at 40 cubic feet per minute, which is over 1,000 liters per minute. So it's a high volume, very high volume sampler. And the one electrostatic sampler was developed at UNC Chapel Hill. And then in the third group, we, in addition to the Anderson impactor, we had 3 additional impactors, a multi-orifice uniform deposition impactor from a local company, MSP Corporation, although they've now been bought by TSI Incorporated. And then one from the University of Minnesota that was a development instrument referred to as a trichotomous virtual impactor. And then one, a Series 230 high-volume cascade impactor. So again, we did these in 3 separate sets. I'm going to be focusing mostly on the results in set 1, group 1, because we haven't fully gotten through all the analyses of the data from the other 2, although I'll show you a little bit of that data. So some of this, I've been working on the data yesterday, so it's pretty new. Our methods for this test, we used actually 3 different viruses, including H3N2 swine influenza virus, which is what I'm going to focus on here since this is a swine conference. We also have an avian influenza virus and a bacteriophage, which is commonly used in many virus tests that we worked with. But we grew H3N2 swine influenza virus in MDCK cells, grown in Eagle's minimal essential medium with supplements. We added a fluorescein dye to our suspension in order to track our physical collection efficiency, because the viruses can potentially break down or become inactivated, so we wanted to have something that helped us measure our physical collection efficiency. The virus suspension was aerosolized at a pressure of 20 psi using a nebulizer, a 6-jet Collison-type nebulizer, in an isolation room at the St. Paul campus of the University of Minnesota. And so we collected simultaneous samples among each group or set of samplers for 30 minutes. And we did that for each set of samplers 3 times. And the suspensions that we got from those samplers were analyzed in different ways. We titrated the samples to determine the quantities of live virus. We extracted viral RNA for QRT-PCR to determine quantities of total virus, and then we measured the fluorescein dye concentration using a spectrophotometer. We can calculate something called the relative recovery, which is basically a ratio between the live virus from the titration and the physical collection efficiency from the fluorescein dye to give us a relative recovery, basically the fraction of the virus that stays live through our sampling process, through both the aerosolization, traveling through the air until it reaches our sampler, and the sampling process. So our isolation room looked like this, and I forget the exact footage of it, but it's maybe approximately 8 feet by 12 feet long. It's not a very big space. On one end, there's air coming in, and in front of that, we placed our nebulizer. You can see it's actually nebulizing there. In this, when we took these pictures, it wasn't producing virus. It was just nebulizing a blank liquid. And that produces the aerosol from high pressure from a compressor, supplying the air here, and then generating the aerosol in front of this supply air that dispersed it into the room. And we measured the uniformity of the concentrations and found that at the other side of the room from where I was taking the picture, we could place these samplers and be relatively uniform in our concentrations at these locations. And this is the set 1 samplers. This is the Bobcat sampler, which is the filter one. This is the SpinCon, which you can't really see what it's doing, but it has a case to make it relatively portable. This is the Vivas sampler from the University of Florida, our AGI-30 and our biosampler here connected to pumps that are on the floor. This is our NIOSH sampler. This is the cyclonic collector. And there's our Anderson impactor right there. So the Hamada and Adepeju managed these tests and ran these all together. I should mention that this room no longer exists. They tore down the isolation building to put up a new one. Well, the new one's already up, and they tore this one down subsequent to our tests. So this space doesn't exist anymore. So now I'm going to move into some results, try to move through these relatively quickly, most of which focus on set 1. So what you'll see here on the vertical axis, in this case, is the geometric mean of the 3 replicate tests of each type of collector for swine influenza virus, and the titer in TCID 50 per milliliter, tissue culture infectious dose 50%. And then you see the 8 samplers here in set 1, and the error bars represent the standard deviation around the 3 tests, and then we did statistical tests to see which was different from each other. First thing you can see here is that probably there's about a log and a half, a factor of maybe 30 difference between the low and the high as far as the titer collected in the samples. But that amongst many of the collectors, there's not a large statistical difference. The SpinCon had overall the highest geometric mean, but it was only statistically different. The A, anything with an A, is statistically not different. So the SpinCon was only statistically larger than the NIOSH sampler and the biosampler. as far as the live titer. So there's not a large difference among many of these samplers for titer. So this is the amount in the liquid into which these samplers put the virus. So if we look at multiplying that by the total volume of liquid, we get kind of similar results. Again, the SpinCon and Bobcat commercial samplers that sampled with very high volumes of airflow got the largest titer. Some of the others, the NIOSH sampler in particular, that had the lowest flow rate, had the lowest total virus sampled. So some of that kind of makes sense. Another way to look at this is to look at the air concentration measured by these samplers. So we know how much was collected. We know how much air volume they processed. So how does that reflected in the air concentration that they measure? And so you get a different ordering of the devices based on this metric. And you get the impingers, the AGI-30 and biosampler, and the VIVAS, which is also an impinger, showed the highest concentrations, and the SpinCon and the Bobcat, while not statistically different, generally had lower concentrations measured. So depending on what you're interested in, whether you want to maybe detect the virus at all, or whether you want to maximize the concentration, may give you different results. So this was all done based on live virus. If we look at the total virus observed from PCR, you get kind of similar results. This is, again, the total virus observed in our samples. And here, again, the SpinCon was the highest. And it was significantly larger than several of the other samplers as far as its ability to detect any virus at all through its RNA copies per milliliter of sample. But when we look at the concentration in air, we see kind of the same ordering as before, where the AGI-30 and the biosampler and the VIVAS gave us the highest airborne concentrations, and some of the others that were the commercial instruments gave us lower concentrations, even though they gave us higher levels of virus observed. And then we can also look at relative recovery, and basically this tells us, if you look at these numbers, this tells us that somewhere around, at the most, we get about 0.1% of the virus that we expected based on if all the virus stayed live through the transmission through the air and then the sampling process. Only about 0.1% at the most stayed live and infectious. So a lot of the virus becomes inactivated as a part of being in the air or being sampled. And these are mechanically generated aerosols, though. So they're not in respiratory fluids. They're not maybe protected in ways that they might be in the real world. When we compare to some other, to the other sets, so this is the group 1, this is group 2 with the filter samplers, and group 3 looking at the virus observed, RNA copies per milliliter of eluent from the samplers. We see that generally, there are some samplers that are similar to the SpinCon, but not really greater than the SpinCon. In particular, the high volume sampler and this trichotomous sampler, which also sampled at a very high volume, gave us similarly high levels of virus observed. So the high volume samplers, the high flow rate samplers, gave us the highest amount of virus per milliliter of sample. So not too surprising there. And again, with air concentrations, several of the samplers, including the filters, the glass fiber filters, the gelatin filters, as well as one of the other impactors, the Moody impactor, gave us pretty high levels of concentration, but not the high flow rate. Samplers don't seem to give us high concentrations. So it's kind of an interesting difference. So what's this all mean? I don't know entirely, since I'm just kind of starting to sift through this data. But what I've kind of observed so far is the high flow rate samplers tend to yield higher titers, more RNA copies. The high flow rate samples consolidate the sample more, so that you bring a lot of virus into a smaller volume of liquid, so that's probably why you're able to see more in your sample. So these high volume samplers are likely to be better for detecting whether there's any virus at very marginal, low concentrations. However, the highest airborne virus concentrations are observed among lower flow rate samplers. So maybe that consolidation process might affect our ability to keep virus live or keep it undamaged so that it can be detected through PCR. I showed RNA copies in comparing across the different sets. We haven't really, we're not ready to show the live virus results yet, but what we can see so far suggests that the impinger samplers are likely to do a better job of keeping the virus live than filter samplers and impactors generally. Ease of use is important. So one of the samplers that is used commonly in the field by the University of Minnesota researchers, the cyclonic collector, doesn't really show very well either in the virus titer or in the concentration measured. On the other hand, it is super easy to use compared to many of the other samplers. So what's the balance between ease of use and getting the best results? Ideally, you have both somehow in a sampler that we develop. Ultimately, a two-sampler strategy might be best. Maybe having a high flow rate non-sizing sampler to detect whether there's anything there that's live or anything that can be found in the air. And if you do, then maybe using a lower flow rate size separating sampler for concentration measurements. And that's similar to some other strategies in occupational health that we use for assessing human exposures. So I wish I could come here and tell you, yes, we know exactly what the best sampler for you to use is. Unfortunately, I don't have that answer. Because there is no one best sampler, a recommendation might be based on your particular application. Hopefully we can develop a better sampler. That's what we're working towards, or a combination of samplers. So our next steps are to compare several of the best performing samplers in field tests this coming flu season, then design and build a novel size separating sampler, maybe one that could be used as part of a two-sampler approach. So maybe we might use one of the existing high-volume samplers together with one that we develop, and then compare that in the field. So I want to acknowledge, again, I mentioned NIOSH is our funding agency for UMass.